Next speaker is Yongqing Yang from New Jersey Institute of Technology. Ultrafast microdroplet digestion of antibodies with Fc silencing mutations.
Hello, good morning, everyone. Thank you for joining my talk. My name is Yongqing Yang, and today I'm very delighted to talk to you on how we perform the ultra-fast microdroplet digestion of antibodies with Fc silencing mutations. This work is a collaboration of NGIT, Agilent, and Johnson & Johnson. The authors declare no competing financial interest. So first of all, let me introduce you to microdroplet reactions. Microdroplet reactions occur on 10 micrometer or smaller droplets, which could accelerate the reaction speed by 1,000 to 1 million folds compared to the bulk solution. And for some microdroplet reactions, the reaction time are even less than 200 microseconds, which is super fast. Next, let me introduce you to antibody digestion and the characterization workflow. So for the intact antibody characterization workflow, which shows in here, it's very simple. What we need to do is only to transfer the sample into the small vial and then use the LC in combination with the mass spec system to do the characterization. But for the typical antibody digestion and the characterization workflow, it's very time-consuming. The first step is to use the enzyme to digest the antibody. This step normally takes 1 hour or even longer. And then we will transfer the digested antibody to the small vial and then use the LC in combination with the mass spec system to do the characterization. Therefore, in order to make the workflow shorter, we designed an flash characterization workflow. What we need to do is only to transfer the sample into the small vial and then use the autosampler in the LC system to draw the sample and the enzyme and then mix them together. And through flow injection and analyze to inject the sample into the mass spec. And in here, the microdroplet reactions happens in the ESI source in microseconds, which enabled by using the Agilent jet stream source. We will discuss this part later. So by using microdroplet reactions, antibody could be digested with IDUS. We already published one paper in analytical chemistry in this area. The workflow shows in here. So the first step is to use 2 different syringe pumps to introduce the IgG and IDUS separately and then mix them into the tea, and then spread it out by using the high-pressure nitrogen gas. And after collecting the sample, we will use the LC in combination with the mass spec system to do the characterization. So the result shows in here. After microdroplet reactions, the antibody could be digested with IDUS within only 50 microseconds. And in here, the manual sample introduction setup time is close to 20 minutes, which is shorter than the typical methods. And then we designed an automated online microdroplet reaction workflow, which enabled by the Agilent jet stream source and autosampler. And in this method, we only need 2 minutes for the sample introduction. Recently, our collaborators, such as Harsha Guanwandina from Johnson & Johnson and Mike Neiman from Agilent, have performed ultra-fast, high-throughput microdroplet digestion by using rapid 5-blaze system, which only 6 seconds for the sample introduction. This animation shows how Agilent jet stream source works. When the jet stream source off, we can find a lot of samples lost when spread it out, and the signal-to-noise ratio is comparatively low. When we turn on the jet stream source, we found that most of the samples are getting to the mass spec, which is good for the characterization, and the signal-to-noise ratio in here is very high. And the high temperature of the jet stream source could help to form the smaller and micro-sized droplets, which is good for the antibody microdroplet digestion. The right picture shows the CFD simulation results, which done by Agilent. the simulation result shows that the velocity of microdroplets in here is close to 150 meters per second. And the estimated microdroplet reaction time in here ranges from 20 microseconds to the 1 milliseconds. Next, let me introduce you to the antibody Fc silencing. In some antibodies, the Fc region could be recognized by effector cell and the complement, which might cause some bad results, like ADCC, ADCP, and CDC. By introducing various mutations at the Fc region of the antibody, we can find the ADCC, ADCP, and the CDC mechanisms will be abolished. What we call the Fc silencing here of this phenomenon. And the Fc silencing is very good for the therapeutic goal. However, for the Fc mutated antibodies, it's very hard to be digested and characterized. For the wild-type antibody without any mutations, we normally use IDUS to digest them first and then to analyze the subunits. But most antibodies with Fc silencing mutations are resistant to IDUS digestion and subunit analysis. Recently, Genovis created a new enzyme which called Fabricator Extra, could be used to digest Fc mutated antibodies. In here, for the first time, we performed rapid online microdroplet digestion of Fc silenced map with Fabricator Extra enzyme. And we already published this paper in the Analytical Chemistry. So first of all, we used two tool antibodies in here. One is the wild-type antibody, another one is the LALA-DS mutated antibody. The reason why we choose these two tool antibodies is to evaluate the digestion efficiency of antibodies which sequences are identical, except LALA-DS mutations. And in here, we performed both IDUS and Fabricator Extra to digest both antibodies under both in solution and microdroplet conditions. And as we know, the cleavage site in here of IDUS is between 236 glycine and 237 glycine. But for the Fabricator Extra, the new enzyme has two different cleavage sites. One is the same as the IDUS. Another one is between 235 alanine and 236 glycine. Before we show our results, I want to introduce you how we calculate the digestion efficiency. The formula shows in here. The I1 is the deconvoluted mass spec signal of intact antibody. The I2 is the deconvoluted mass spec signal of antibody after digestion. Therefore, in this example, the digestion efficiency is 87.6%. So for wild-type antibody, the tool antibody, we performed both IDUS and the Fabricator Extra to digest it, and the result shows that both enzymes could digest it under both in solution and microdroplet conditions very well. But for the LALA-DS mutated antibody, we showed in here is a totally different story. To see the scheme on the top, it shows the subunits which will be formed after the intact LALA-DS mutated antibody digested by Fabricator Extra. We first obtained the intact antibody mass spec signal in here. And then we performed IDUS to digest the antibody. The result shows that IDUS cannot digest it at all, no matter on the in solution and the microdroplet conditions. And then we used the Fabricator Extra to digest the antibody under both conditions too. And the result shows that it could digest the antibody with 80% digestion efficiency under both in solution and microdroplet conditions. So in order to improve our subunits mass spec signal, we designed a new strategy here. The first step of this new strategy is to use TSAP to reduce the double chain antibody to the single chain. The result shows the reduction happens very successful. And then we will use the Fabricator Extra to digest the single chain antibody to the subunits. Since the single chain antibody structure is much simpler than the double chain, therefore it will only form two different subunits, and the subunit signal might be higher. Our results confirm the hypothesis that the subunits after after the reduction signal is very high. And we found that after reduction, the single-chain antibody digestion efficiency is higher than 90%. So in addition to the in-house made two antibodies from Johnson & Johnson, we next looked at some commercial antibodies with FC mutations. The first is vedolizumab, which could be used to treat the inflammation and has the LAGA mutation at the FC region. The result shows that idols cannot digest it at all, no matter under in solution and the microdroplet conditions. But the fabricated extra could digest it very well under both conditions with 85% efficiency. And next, we looked at anti-PD-L1 and anti-PD-1 antibodies, which are very crucial to kill the tumor cell. The left picture shows that PD-L1 receptors are found on tumor cell, and the PD-1 receptors are found on T cell, which could combine and to form an immune synapse, and then will inactivate the T cell to kill the tumor cell. The right picture shows that anti-PD-1 and anti-PD-L1 antibodies for T cell mediated tumor cell killing by PD-1 or PD-L1 blockade. For the livolumab, the anti-PD-1 with LF-LE mutations at the FC region, the result shows that both idols and the fabricated extra could digest it very well, no matter under in solution or microdroplet conditions with very high efficiency. But for the LALA-PG mutated anti-PD-L1 antibody, the idols cannot digest it at all. But for the fabricated extra, we can find the result shows that under both conditions, it could digest the antibody. But we should notice here, after 1 hour in solution digestion, the intact antibody signal are still very high, which means the digestion efficiency is comparatively low. Then we compare the digestion efficiency of this kind of antibody. The left plot shows that the idols cannot digest it at all. And for the fabricated extra, after 1 hour in solution digestion, the efficiency is much lower than the microdroplet conditions. The right plot shows the digestion efficiency as a function of in solution digestion with the fabricated extra. The result shows that after 2.5 hours in solution digestion, the efficiency is close to the microdroplet conditions, which only needs less than 1 millisecond. In order to make our workflow much more accessible to the industry scientists, we designed an automated enzyme to antibody selection scheme and performed online microdroplet digestion in this work. The workflow shows in here. The first step is to use the correct enzyme digest the antibody based on the presence and absence of mutations on the sequence. Therefore, we will use a script to read the sequence of antibody and pair the appropriate enzyme to antibody digestion, and then create some methods. And these methods could be imported to the Mass Hunter software to form a work list. For each method here, we program injections. Yeah, so for example, for the wild-type antibody, we will only choose idols to do the digestion. For the LALA-DS and LALA-PG mutated antibodies, we will only choose the fabricated extra. And then we will use autosampler in the LC system to mix the enzyme and the antibody. Then through flow injection and the mass spec system to do the microdroplet digestion and characterization, which enabled by the Agilent Jetstream source. And in here, for the high throughput test, we used 94 samples of 3 different kinds of the antibody, wild-type, LALA-DS, and LALA-PG mutated antibodies into the 96 well plate. And we also put 2 different enzyme, idols and fabricated extra, in the well plate too. The result shows in here. The different color of the box shows the different kinds of antibodies. And for each sample, we make sure there is no carryover from previous injection. And the number inside the box shows the digestion efficiency of each sample. So after summarize the results, we draw the box plot in here. The box plot shows that the reproducibility of our methods is very high, with only 2% CV. And the robustness tests, we changed the concentration of mobile phase and auto-sampler temperature. The result shows that only 5% CV difference here. And for the high throughput test, the result shows that for the 94 samples we performed here, only needs 104 minutes, which means 1.1 minutes per sample. So it's very promising to the industry. These are the conclusions we show of this work. First, a few antibodies with Fc silencing mutations are resistant to IDUS digestion, and for the fabricated extra, it could digest the Fc mutated antibodies under microdroplet conditions with a high efficiency. For most of the antibodies we tested here are higher than 80%, and the reproducibility in here is still very high. And for the LALA-PG mutated PD-AL1 antibodies we showed here, it could be digested under microdroplet conditions, and the efficiency is much higher than the 1 hour in solution digestion. And for the automatic injection workflow we designed here, the high throughput test shows that it's very promising to the industry. Finally, I want to thank all the members in the Chen Lab, especially my supervisor, Dr. Hao Chen. And we want to thank all the collaborators, such as Agent Technologies, Genovis, and Harsha Guanawandena from Johnson & Johnson. We want to thank all the funding agencies. And finally, I want to thank you for your attention again. Thank you for your listening. Yeah.
Thank you, Yongqing, for the great presentation and very nice work for the high throughput analysis. We have a few minutes for some questions, and please come to the center. Yes, please.
Nice talk and great technology. When we cleave below the hinge, we have 100 kilodalton piece. So complexity was not decreased significantly. If we do digest limited proteolysis above the hinge, we get 50 kilodalton, 50 smaller pieces. What the advantages of cleaving below the hinge?
Yeah, sorry.
Versus above the hinge, what the utility versus cleaving above the hinge?
Yeah, we are cleave under the hinge.
Yes. Why not above the hinge?
Why not above the hinge?
Yeah. Because complexity is still relatively high. On the top, you have 100.
Yeah.
It's not 150 kilodalton whole antibody, but it's still 100 kilodalton on the top, right? And
yeah.
No, anyway, you may consider cleaving above the hinge, a smaller piece easier to handle after. Thank you.
Okay. Thank you.
But otherwise, rapid, reactions in microdroplets, it's quite impressive. Thank you.
Okay. Thank you for your suggestion. Yeah. Thank you so much.
Yes, I think for Pavel's question, it's more like if you can cleave above the hinge, you generate like 350 kilodalton versus 150 kilodalton and 100 kilodalton.
Yes.
Right. Very great enzyme selection here, and also I actually have a question. So, is there a way to combine the fabricator extra with the reducing reagent and then do?
Yeah. We also performed this experiment, but we not show here. So we just use the autosampler to draw the antibody, the TCEP, and the fabricated extra, and then mix them in the LC system, then do the flow injection, and the result is also very real. Shows that like 80% of the antibodies will be reduced first, and then in the microdroplet, the fabricated extra could digest the reduced antibody with also very high efficiency.
I see. And also, my other question is there another chance for other enzymes to be, implemented in this type of technology? Like
Of course, yeah.
Did you?
Yeah, we already tried some bunch of different enzymes, and most of them are works.
Most of them, okay.
Yeah.
Great. Any other questions? Okay. Let's thank Yongqing again for the great presentation.
Yeah, thank you for your attention again.